Good morning, everybody. I'm going to try to hide the Boston accent, but welcome. I'm probably one of the only Boston-born boys in the room today. So, on behalf of us, enjoy the great hot and humid weather that we have. 9 years ago, I started this odyssey with advanced stage 4 renal cell cancer, and as the girl dad of 4 daughters, I was not sure if I was going to see all 4 graduate high school, all 4 graduate college, let alone get engaged and get married. And I'm happy to report as of a month ago, I've crossed off all 4 high school graduations and all 4 college graduations. And thank you. My first son-in-law survived the "can I marry your daughter" discussion. None of that would be taking place without the amazing work that each of you do with your organizations. And I feel that I'm the poster child or the poster boy of all the great work that each of you and your teams do. But the only way we're able to pull that off successfully is with funding. And it scares the heck out of me that when my fight started in 2015, there were 0 federal dollars allocated towards kidney cancer research. And thanks to all of the great work of Brian and Deb and Susan and the KidCAN team, we have changed the paradigm. And I know this year we raised $50 million. But I know, as someone who's exhausted all standard of care, that my pal Tony Sherreri needs more tools in the toolbox as my cancer continues to morph and adapt. And when this trial stops doing what it's supposed to be doing, my hope is that my buddy Tony has something else in his toolbox that we can use. But the only way we can do that is through research. 5 years ago, at the Dana-Farber Harvard Cancer Center Symposium, I challenged the Dana-Farber team, the BI team, and Dr. Michelson's team from MGH to join us on advocacy days. I've seen firsthand the power of what these advocacy days do. And there's nothing better when the patients and the alliances get together. But as a sales and marketing guy from my career, I knew we were missing something, and we were missing the clinicians and the scientists. And 5 years ago, I challenged the local team to join us. And over the last 4 years, whether it's been Dr. McGregor, Dr. McDermott, Dr. Henske, Dr. Mahoney, and their researchers, Renee and some of the other folks, they've joined the call, they've joined the fight. And my takeaway for each of you today is, help us help you. I need your help. My 4 daughters need your help. My wife and my golden doodle need your help, because there's a whole bunch of stuff that I still want to check off my checklist. And the only way I'm going to do that is with your help. But the only way you're going to continue to do what you're doing is if we get more funding. So please, September 24th, we're doing it live in DC. Anybody that wants to join us down there, by all means you can. But we need 20, 30, 60 minutes of your time on the 25th to join us. It's really simple. We actually, believe it or not, anybody wearing the orange badge, orange lanyard, we will train you. We have training sessions for how to talk the advocacy piece. So I have a challenge for each of you. I need you to go back. I need you to register your teams. It's very simple. We have a QR code. So during the first break, I'm going to be in your face as you're coming out the door. I want each of you to take one of these. I want you to take it back. And when you're on the plane tonight, your first assignment is do the QR code, sign yourself up, and then bring it back and sign your teams up. Because without your help, without continued advocacy, we aren't going to be successful. And again, I'm not here without the great work that each of you and your teams are doing. But think about how much more we can get if next year we get 60 million or 70 million or 75 million. So that's it. At the first break, I'm going to have a ton of these. Please take one. When you're on the plane, when you're sitting at the airport, please register yourself. But more importantly, ask your researchers, ask your nurse practitioners, anyone that has time on September 25th. Mike Sherjan knows, Dr. McGregor knows how easy these calls are. And as someone who's done this now for the last 5 years, our government officials like nothing better than listening to the patient story, and more importantly, listening to you and what the latest research trends are, what the latest developments are, and how you're putting their money to use. So please, 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 9/24 and 9/25, our advocacy days. I need your help. Thanks, everybody. I'm going to kick it off now with Tony. You're next. Yep. Oh, Hans. So, and again, enjoy your time in Boston, everybody. The Red Sox are on a roll, so anybody going to the game tonight, you should really enjoy it. So, it's all over you. Thanks. You got it.
Well, if that is not inspiring, so I have a great honor to introduce a real treat. I had to apologize to Dr. Rathmell that there will be no hot and heavy bro pictures like Tony yesterday had with Charlie Swanton, so I apologize. It's going to be a little bit more formal, if you will. So our next speaker is Dr. Jeff Rathmell, and he is really well known for, I think, a very exciting topic, which is immunometabolism. There's a very particular interest in T-cell fate, association with metabolism, and he started his training in Iowa, went then to Stanford, got his PhD there, and then trained actually with Craig Thompson, so one of the greats in metabolism, and then started off his academic career in Duke, went to Vanderbilt, is the Cornelius Vanderbilt Professor of Immunobiology. As you know, he is part of a power couple and has published on that, so very exciting. His wife is a great friend of kidney cancer. He is very collaborative, very well funded. He takes mentorship seriously. And today we are in for a real treat. We're going to talk about the relationship of obesity and immunobiology. Thank you so much.
Yeah, well, thanks for the introduction, for the opportunity to be here and talk a little bit about our work. So I'm going to talk about obesity immunotherapy, but it's very much a basic science perspective. So I'm not actually going to treat any patients. We're going to treat some mice. Okay. So here's my disclosures. It's actually on the board of Citrix Therapeutics. It's not actually relevant for today. All right, so we've been interested now for, it's actually almost 25 years in trying to understand the metabolic transitions that happen when cells are stimulated to grow and proliferate. So if we look at just like mostly we use T cells, but the model is very similar for cancer cells and many other settings. Cells are a quiescent resting mode, or like a T cells in surveillance. It needs a lot of ATP to do that, to be moving around and looking for its target, but it doesn't need to build or anything. So it's a lot of burning fuel. It's catabolic metabolism. And when cells are stimulated, neoantigen in cancer, or pathogen, autoantigen, will grow and proliferate, and of course they have to switch to an anabolic metabolism. Taking up nutrients from the microenvironment allows them to build. And we showed, actually, again, this goes back to one of my first papers that when I was in Craig's lab, you mentioned. It shows that when lymphocytes are stimulated, they upregulate nutrient transporters. In this case, it's GLUT1, it's a glucose transporter, so they can take up nutrients. And then that, of course, forms the basis for positron emission tomography, or PET imaging, which is used, of course, widely in cancer, obviously to monitor progression and diagnose. So, of course, this is an image of a non-small cell lung cancer patient, and of course, the tumor takes up the FDG, the fluorodeoxyglucose, but obviously other organs like brain uses glucose, so it's not just the cancers. And if you look at inflammation, this is actually arthritic joint here on the right. The arrows are pointing to some bone lesions, and you can see those lesions are also taking up glucose. So inflammation is also a highly glucose-consuming process. But this is a very high level. This is just at a whole tissue level, and this is a little unsatisfying to us. So one of the things we did now a couple years ago was we went and decided, well, let's look at what are the actual cells that are taking up the glucose. And the way we did this was we just took animals here, we gave them tumors, and then treated them with the fluorodeoxyglucose, the same compound that goes into the patients. And then you can remove the tumor, fractionate the cells, and see which cells actually take up the glucose. And what was surprising to us was it's not actually the cancer cells. It's actually more the inflammatory cells. The cancer cells are using glucose, so it's not like there's nothing there. But if you really break this down, the immune cells, and really this break down to the macrophages predominantly, they are the primary glucose consumers. And this was across a wide variety of animal models. The cancer cells are actually really good at taking up glutamine and lipids. So the metabolism really has to be defined, not necessarily at a tissue level, but at an individual cell level. You have to think about what all the different cells are doing. Okay, so now if we go back to this model and we think when T cells get activated, they grow and proliferate, Now again, you have to think about what are they doing individually, and when T cells get activated, they differentiate into different subsets. So they all take on different characters. So there's a wide variety of cells involved here. And generally, they fall into a few categories: Treg, Th1s, Th17s. The myeloid cells, the macrophages, have their own sort of divisions here. And it's much more of a continuum in the myeloid cells, but it's a number of populations. And we did show now, a number of years ago, that when cells take on these different characteristics, they also take on different metabolic programs. So it's not just the glucose uptake from the PET imaging. The whole metabolic program can differ. So this is metabolomics, so this is the metabolome, naive T cells, Th1s, 2s, 17s, Tregs. You could do the same thing with myeloid cells, stimulate them different ways, you'd get the same different profiles here. And the key thing are, the naive cells, these are quiescent. Everybody else over here, the Th1s, 2s, 17s, they're all proliferating, but their metabolism is all different, so that anabolic program differs. And it's a little overly simplified to generalize it into one basic model, but the general trend seems to be that cells that are more on the OXPHOS level, a lot more mitochondrial genes, they tend to be a little more suppressive, and the cells that are more on the glycolytic end of the spectrum tend to be more inflammatory. That's sort of just a general rule that seems to be the case. All right, so now with that sort of in mind, I want to jump in and talk about how obesity affects cancer and some of these principles and how we think that apply. Right. So everybody is going to be very familiar with obesity as a sort of growing epidemic, and you've seen those charts as rising rates of obesity across time in the country. But it's also important to think about the disparities aspect of this. It's not just that there's more and more obesity, there's more obesity in specific populations stand out. So this actually shows the racial and, excuse me, let me go backwards, there we go. So racial and ethnic background, and show that it really is quite variable. So we need to think about this at a lot of different levels. All right. So if obesity is increasing, it's associated with all sorts of different diseases, all sorts of different pathologies, and cancer, of course, being one of them. And this is AACR data on the left side here. The highest modifiable risk factor for cancer is smoking, but if you go immediately next to that, you've got physical inactivity and diet and alcohol. Many of these things that tend to go together, just sort of poor lifestyle. So this is actually one of the, cumulatively, one of the most important risk factors for cancer. Now, it's not that every cancer is at risk here. There's some that have quite a bit more clear-cut risk. And the NCI has designated 13 as sort of obesity associated. There's a few others that have trends that way, but these are the most clear-cut cancers. And actually, one of the things that's also sort of interesting is weight loss can protect in some of these cases. And there was a nice study in JAMA that just came out pretty recently showing that the Ozempic, the GLP-1 agonist inhibitors, can protect against cancers. But interestingly, they don't protect against all of these 13. It only had effects on 10 of the 13. Right, so it's kind of, so there's, you know, what's different about those other 3? So that's going to be, and kidney cancer was affected by that, but it wasn't dramatically affected. It was sort of a modest effect. All right, so anyway, so obesity, cancer. So how does obesity actually then drive cancer? And there's a couple ways to think about it, and there's multiple mechanisms. There is a direct effect between the adipocyte and the growing sort of hypernutrition, a direct effect on the cancer cells. And there's multiple mechanisms for that. It can be the nutrients themselves, can be the hormones, insulin, leptin, et cetera, can directly affect the T cells. But if we think about the T cells and the macrophages that are in the tumor microenvironment and just the general role of the immune system here, it's a lot less clear. Now we know that obesity mediates a lot of its functions, its pathologies, due to this chronic inflammatory setting. So then the question is, well, how much does this change in inflammation actually affect the tumor? And might this be driving or contributing? So there's been some work on this at sort of the single cell level, and I'm going to talk about some of our data that's parallel to this, but I wanted to highlight this one paper. This is from Marcia Hagis's group, came out a few years ago. Alison Ringel is the first author. She's now has her own lab at MIT. And what they did was basically a single cell RNA-seq experiment looking in lean and obese, mostly animal models, but they did some human correlates to back it up. And what they found was that the cancer cell metabolism in an obese setting did change. It upregulated more lipid metabolism, and that then changed the microenvironment in a way that actually directly suppressed the T cell. So the T cells were directly changed by this altered cancer metabolism. Right. So our data matches this, plus I think we have a little bit different add to it. All right, so one of the ways we started to get at this was just, again, just an animal model to get us going. So this is MC38, it's colorectal cancer. We gave this to animals and put them on low-fat or high-fat diet, and then they're treated with PD-1 or immunoglobulin control. And what you can see here is that the high-fat diet mice, the tumors grow much faster than in the low-fat diet. But if you treat with PD-1, you actually get quite a good response in the high-fat diet mice. Actually, a greater response in the high-fat diet than in the low-fat diet. So this has been called the obesity paradox, and this was shown nicely in this Nature Medicine paper about 5 years ago. And basically the premise is that obesity is a risk factor for cancer, and so it can promote cancer progression, but it can also sensitize to immunotherapy, right? So you can get this sort of double edge. You can do better in one sense, but worse in another, right? So what is the mechanism of that? And this is now our data. We did the same single-cell RNA-seq sort of approach that Marcia's group did to start with. So we have our low-fat diet, high-fat diet, same tumor, MC38 plus minus PD-1. The first thing we did is we sorted the cells into CD45 negative fraction, so basically the cancer cells or the non-immune cells, and then we also sorted the CD45 positive. So I'll show you the CD45 negative here. So this is the UMAP where you can see where we've identified the different populations here and a heat map of the abundance of those cells below. And the key thing here is if we look at the high-fat diet versus the low-fat diet, just in the control case, it's pretty similar. We don't actually see a big change in the populations of the CD45 minus cells with just the diet. Now this large island down here, that's the epithelial cells. We think those are the cancer cells. Now if you treat with anti-PD-1, you do get a good response and the tumors are starting to shrink. So we're actually losing those cells and so they're going away. So there's probably some differences here, but we haven't really dug too deeply into it because it's nothing really jumps out. All right, so I'm going to talk mostly about the CD45 positive fraction, the hematopoietic compartment in the tumor. So again, this is all the populations identified at the top, with the T cells on the right islands there, then this big monocyte macrophage island here in the middle. And I'm going to tell you a little bit about the T cells, but the thing that really stands out is this low-fat diet versus high-fat diet, there's a huge change in the myeloid population. The macrophage population within the tumor changes dramatically in an obese setting. Now the T cells do change as well, it's just harder to see here, and so we looked at that, and our data matches almost exactly what the Haggis lab had shown. So this is we sequenced the T cell receptors as well to get an idea of the clonality and how expanded are the T cells. So that's what this is shown on the left, but it's really quantified here on the right. This is low-fat diet, high-fat diet. So the yellow on the top, those are sort of the hyper-expanded clones. There's fewer of them just with high-fat diet. So the cells are not as expanded in the obese setting. But as soon as you treat PD-1, they sort of just jump off and they get going, and you get this nice increase in T cells and the increase in these highly expanded clones. So the T cells are less activated or less expanded to begin with, but they can respond. And you see the same thing if you look at granzyme B. It's just a marker for effector function. It's lower in just the basal high-fat diet state, but it bounces back pretty quick as soon as you give the PD-1. So the T cells are less activated, but they can respond. Right. So one of the hypotheses as to why this could be the case is that PD-1 itself might be suppressing the T cells in the tumor, and maybe they actually have more PD-1. And the Nature Medicine paper I showed you earlier did actually see that. They said they had a higher PD-1 expression on T cells. So we looked at that. So is PD-1 actually itself contributing to this? But we don't actually see any difference, excuse me, don't actually see any difference in the PD-1 expression on the CD8 T cells. And if anything, there tends to be a little bit of a trend downward in the number of models that we looked at. So it's not like the T cells are expressing more PD-1. But what we did see that was a little surprising to us was that the tumor-associated macrophages, that increased macrophage population, they started to express more PD-1. So they're starting to induce PD-1 in the obese setting. Now, one of the things that stands out is they don't express near as much PD-1. So if you do the comparison of these histograms, this is the same experiment, so you can actually look vertically here. They do express some PD-1 compared to the low-fat diet. The high-fat diet shifts to the right, but it's still a lot less than what the T cells have. So it's kind of easy to think why people didn't really pay a whole lot of attention to this. It's pretty subtle. So this is in the MC38 model. This is a kidney cancer model that we got from Hakimi. This is the LRVC C67, and just basically shows the same thing. In a high-fat diet setting, these tumors grow faster than in the low-fat diet. And if you look at PD-1 expression, PD-1 is higher in those tumor-associated macrophages in the high-fat diet setting. No change to PD-1 on the CD8 T cells. So it seems like the T cells are not responding to the diet the same way the macrophages are. Okay, so we're not the first one to see PD-1 on macrophages. This has been actually known for a while. So there's a really nice paper from our Weissman's group back in 2017 even, where they showed that PD-1 was expressed on tumor-associated macrophages, and they used some blocking antibodies and showed that this could affect phagocytosis and ultimately the tumor itself. And then Vicky Busiotis from here, she's up in Boston here, so we collaborated with her on this project. She showed in 2020 by using a PD-1 floxed animal and a myeloid Cree, so she could knock it out specifically in the myeloid cells, that those tumors don't grow very well at all either. So again, genetic evidence that PD-1 can play a role in myeloid cells. Right. So what about human tumors? All that's been mouse so far. So this is kidney cancer patient data. This is the same patient here, just one example here. CD11B, so these are the myeloid cells, the macrophage population, or monocyte population. PBMCs versus tumor-associated macrophages from that same patient. You see it's a shift in PD-1. Again, it's not a dramatic shift, but it is a real shift. It's just not a lot when you compare it to what the T cells are expressing. T cells express a lot of PD-1, but macrophages have it. And this is immunofluorescence from a tissue microarray. We've done this with Scott Hake, who's here. And we can see that we have T cells and macrophages both positive for PD-1 in these tumors. That is, of course, protein data. Here's some RNA-seq data. This is from single-cell RNA-seq data. Human RCC, we see that the detection is not as good here. Again, they don't express a ton of this. But normal macrophages, normal kidney macrophages don't express PD-1, but in the case of a cancer, we can get some PD-1 expression there. It doesn't seem to be related to the tumor grade. And when we break this down based on the tumor status, that doesn't seem as relevant. It seems to be much more related to the obesity and the metabolic health. Okay, so what are all the things that might be driving this PD-1? And we think it's in essence, it's the inflammation. We think it's the inflammatory drivers of this. And here's a little bit of data to show you, put it in context and give you some feel for why we think this. So this is actually tumor-free animals on low and high-fat diet when we just look in different tissues. So the bone marrow, again, the high-fat diet here versus low-fat diet. We don't really see any PD-1 in the bone marrow or in the spleen. But in the gonadal adipose tissue, you get a big increase. So in this adipose tissue, we know this is inflamed. This is part of the obesity-associated inflammation. We're starting to get some PD-1 there. Now, if you look at a tumor-bearing animal, we see the same thing. There's no real change in the spleen, but in the this is epidermal fat here, and in the tumor, we're getting a shift up. So this is not like every macrophage in the body. It's in specific sites that we know have at least varying degrees of inflammation. So you can also take bone marrow-derived macrophages and just treat them with these inflammatory signals individually and see what actually matters. So it's not everything that will turn up PD-1, but it's a pretty good range of things. So macrophages actually respond to a lot. So in this case, we've taken these bone marrow-derived macrophages. If they're unstimulated, that's sort of an M0 here. If you give them LPS, sort of a very generic inflammatory cue, of course, it is increased in obesity from the microbiome, actually. But that induces PD-1 pretty strongly. Interferon gamma does as well. IL-4, which drives more of this sort of classic M2 or suppressive macrophage, that doesn't do it at all, really. All the M1 or type 1 inflammatory cues tend to do this. And it's not just the sort of classic inflammatory cytokines. Insulin and leptin, these obesity-associated hormones, and of course insulin is, it's going to go up when you have a diet feeding, and it gets hyperinsulinemia in obesity. But those induce PD-1. And then the other thing that will induce it, actually, that's sort of interesting, is if you just give lipids, you give high doses, in this case is palmitate. As you dose in palmitate, there's this process called lipotoxicity, so it actually starts to kill the cells. But the cells that are surviving at the end are the ones that have more PD-1. So the LPS, the palmitate might be inducing PD-1 or might be selecting for those PD-1 positive cells, but there seems to be a connection. Right. So the signals that are going to be driving this, again, we have a lot of different cues that seem to be able to induce PD-1, and we're actually in the process of doing some CRISPR screens to figure out what are the kinases and transcription factors that are key. But at a high level, we can look at pathways like the mTOR pathway, which gets induced pretty strongly after many of these cues, like LPS will activate the pathway here. And if we treat with them with rapamycin to block the TOR pathway, then PD-1 does not get induced. This is a case where we've given an inflammatory cue to macrophages in vivo, treated the mice with rapamycin, and you see that we don't induce the PD-1. Now, conversely, you can turn the pathway on by deleting P10, and so P10 is a phosphatase that when it's deleted, this pathway will get turned on, and that's sufficient to increase PD-1 in macrophages as well. And this is an in vitro cell line, but we've also done this with a patient who is a P10 deficiency. That person, that PD-1 is just spontaneously high on their myeloid cells. Right. Now, the TOR pathway does a lot of things, but we've been interested in metabolism, so we know that it drives some of these metabolic changes. So we want to show just one piece of data around what are the metabolic changes that are important, because it's also sort of interesting. So all these cells are treated with LPS, so they've induced PD-1, but if you block glycolysis with 2-deoxyglucose, they really don't induce as much. So this glycolytic transition that the LPS will induce that pathway, that metabolic transition is important. So I showed you earlier, all the different cells have these different metabolic programs. There's something about glycolysis in that program that seems to be important here. Now, if you block glutamine metabolism with DON, this is a general glutamine antagonist, it doesn't do anything, nor does etamoxir, which blocks some of the lipid uptake. So there's something special about that glycolytic program that leads to this PD-1 expression. Okay, so now what does the PD-1 actually do for the macrophages? And then the short answer, it does basically the same thing as it does to T cells, right? And it suppresses them. So I'll show you that data. So this is actually now looking at some functional assays. These are macrophages from tumors from low-fat and high-fat diet treated with without PD-1. And we see that phagocytosis is down in the high-fat diet, as is the antigen presentation, MHC class 1 and class 2. And both of those are reversed if you give anti-PD-1. So the PD-1 seems to be suppressing the macrophage ability to phagocytose and present antigen. Now this is, of course, at a pretty high level, because we're just taking the tumors out and treating with anti-PD-1. But you can also sort the cells out and see what PD-1 does. And if you look at the gene set enrichment that happens in this case, if the cells are treated with anti-PD-1, you have this upregulation of all sorts of inflammatory gene sets. We've got cytokines here. There's some metabolic transitions here around lipid metabolism, but all these others have to do with inflammatory processes. So PD-1 is affecting, or does appear to be affecting this. So we can go back to our single-cell RNA-seq data set and try to look for some correlates here. And so in this case, this is the same plot I showed you earlier. Now if we pull out these macrophage populations down here and just highlight the PD-1 positive versus negative macrophages, and then do the same sort of gene sets, what we see is the PD-1 positive have an increase in oxidative phosphorylation, lipid metabolism, amino acid metabolism, have decreased phagocytosis, decreased glycolysis, and decreased antigen presentation. That's at the gene expression level, and you can validate this functionally. So if you sort out the PD-1 positive cells for the PD-1 negative cells, you see that the PD-1 positive cells have high oxygen consumption, consistent with high oxphos here, high lipid uptake, so they're taking up lipids and seem to be able to burn them. If you look down the other side of this, they actually have, the PD-1 positive cells have reduced phagocytic ability and reduced expression of MHC molecules, in this case showing MHC2 there. So that's all, it all correlates with the macrophages being less able to phagocytose and present antigen to stimulate the T cells. But it's all correlative, so you need to go back and directly test this. Now, in the macrophages, it's interesting because they don't induce just PD-1. They also are well known to express PD-1 ligand. So they can have both the receptor and the ligand. And if these are going up in obesity, then there could be some interactions there. But of course, you can take advantage of this with all the different pharmacologic tools. And so we come in with exogenous PD-1 ligand, basically to saturate the PD-1 signal, or you can come in with an anti-PD-1 antibody and block the whole thing. So we can turn it all on or turn it all off and see what happens. And I'll show you just a little data here. So this is when we do this. Now look at the glycolysis, this metabolic transition. And what we see is if you stimulate with PD-1 ligand, so you're turning PD-1 on all the way, glycolysis goes down, it's quantified on the right here. If you block PD-1, it goes up. So PD-1 seems to be suppressing glycolysis. You block that, it immediately can start to rebound and go up. Conversely, lipid uptake does the opposite. So if you trigger PD-1, lipid uptake goes up. You block PD-1, lipid uptake goes right back down. So those metabolic transitions are responding to PD-1, and we think those are important, again, to the function of the cells. In this case, I'm showing expression of just with one example, CD86. This is a co-stimulatory molecule. So if the cells are stimulated here, and then they're given control or anti-PD-1, in the presence of the anti-PD-1 to block that PD-1 signal, they start to express more co-stimulatory molecules. But if you block glycolysis with 2-deoxyglucose, that really doesn't happen. So we think that metabolic transition is coincident and important for the functional change in the cells. Okay, so that's pharmacologic. We can go back and use the conditional knockouts to genetically test this, and then this is in collaboration with Dr. Busiotis. So this is a lysine pre-PD-1 floxed animal, and so this shows that if we get rid of PD-1 in the myeloid cells, the tumors grow more slowly than if they are PD-1 positive. But then we can take those macrophages out and look and see what they're doing. And those macrophages that are PD-1 negative have higher rates of phagocytosis, they actually have reduced lipid uptake. So again, we're turning the cells away from that lipid uptake. And in fact, what they're starting to do is they become very glycolytic. So they regulate glycolysis pretty dramatically here. It goes up at basal and maximal levels. And ultimately, those cells then are far less suppressive and can actually be a little more immune stimulatory. So we have less suppression here, a little more stimulation. And if we look at the T cells from these tumors, they look more activated, but they also look a lot less exhausted. So here we are, there's higher expression of granzyme B, but also reduced expression of PD-1. Here's a quantification, and there's some of the flow plots. So the T cells are less exhausted. So I'm gonna wrap up. This is the model here then, that we think obesity in itself is not necessarily a bad thing, because if we do like just a short time course on a high-fat diet, it doesn't do anything. It really takes longer, to the point where metabolic health becomes impaired. And you have that metabolic inflammation and all the subsequent inflammatory cytokines that can come from that. That then, we think, this inflammatory cue leads to the myeloid cells, it stimulates them, but also then there's a feedback. So the inflammatory signals can drive them to become more inflammatory, but then the feedback with PD-1 then shuts this whole pathway down. Glycolysis and that glycolytic phenotype, big part of that, but also this decreases their ability to drive inflammation and T-cell help. So you get less T-cell activation. So ultimately, PD-1 is kind of doing the same thing to macrophages that you think about it doing to T-cells. And if you think about sort of the universe of immune checkpoints, generally, it's kind of a simple view. You think, oh, there's the T-cell checkpoints and the myeloid checkpoints. I think the reality is it's more of a Venn diagram. There's going to be some checkpoints that are T-cell specific and some that are myeloid specific. PD-1's in the middle. And we haven't looked at this extensively, but I can tell you LAG-3 does basically the same thing. So LAG-3 is also induced in obese settings for myeloid cells. But they're not all, because we don't see TIM-3 expressed, for example. So I think it's going to be interesting to think about how these different checkpoints are relevant in different cells and potentially in different settings, with obesity being one of these inflammatory settings that might be driving this up. And ultimately, we think this is maybe something that's contributing to the obesity paradox, in the sense that in this feedback on the myeloid inflammation that via PD-1 basically reduces the ability of the tumors to immune edit. So you're a little more immune suppressed. So you're not getting that anti-tumor basal immunity. So that might give the tumor, that's going to give the tumor some advantage. Right? So that's going to increase the tumor growth. But conversely, because the same checkpoint that we come in with therapeutically, PD-1, is contributing to the mechanism of the suppression, as soon as you block it, you kind of get you relieve all that. The macrophages can rebound. You kind of get a double. So we think that then might promote this. This paper just came out a couple weeks ago. There's a QR code if you want that. And of course, lastly, need to acknowledge the people who did the work and the team. This is really all done or led by a postdoc in the lab, Jackie Bader. Here's the Rathmell lab. We sort of merged in with Kim's group last fall. So there's the whole team. This was also funded by the Mark Foundation work in addition to our DoD funding. And so this is our Mark Foundation team. And with that, I'm happy to end and take any questions. Thank you. Yes, Les.
So Jeff, I wanted to come back to the comment about the relationship of mTOR to PD-1. Sorry. And a couple of things come to my mind. So obviously there's a link to mTOR and glycolysis through HIF.
Yeah.
So if you upregulate mTOR, you have more HIF, you have more glycolysis, and also hypoxia in particular and HIF to some extent also promotes lipid uptake. So I'm wondering, if you take HIF out of the picture, particularly HIF1, what do you think might happen?
Yeah, no, I think that's a great suggestion. It definitely could be the case, because it'd be interesting. I mean, it's not, in some of these settings, I don't know how hypoxic adipose tissue would get. It could get a little hypoxic.
It definitely gets
hypoxic
under obesity.
So like a hypoxic syndrome could be definitely contributing to this. Like I said, we're doing a CRISPR screen where we're knocking out all the transcription factors and trying to see which of them actually affects the myeloid induction of PD-1. So we'll hopefully get an answer to that, but it's a great suggestion.
If I could have a follow-up, when you say PD-1 is going up, is that at both the RNA level or the surface protein level, or both?
Both. We see both going
So you might imagine glucose is contributing to glycosylation of the protein.
It could be, yeah.
Right.
It definitely could be a glycosylation sort of event.
Great.
Lisa.
I'm wondering if you've looked at the kinetics of these effects on myeloid PD-1. For example, if you switch the mouse from a high-fat diet to a low-fat diet, would these effects be reversible?
Yeah, we have not done that extensively, but we did do it in a one experimental model system, in a breast cancer model system, where the mice were high-fat diet and then they were put on a low-fat diet and they lose weight. And in that low-fat diet setting, the PD-1 went away. The myeloid cells reduced PD-1, so that went back down. So that's reversible. Now, whether that's going to be reversible over what kind of time frame that is, and the weight cycling is what really happens. People gain weight, then they lose weight, then they gain weight, they lose weight, and that weight cycling is not the same as just weight loss. You never really fully return. So what happens in those kind of settings? We're really interested. And actually, there's a question on the screen here about GLP-1 agonists. So how do you think what GLP-1 agonists will impact the findings? We are starting some of those experiments. Definitely, it mimics a lot of the weight loss sort of things, but I have a collaborator, again, part of this Mark Foundation group, Liza Makowski, who's done some work showing that if mice, they gain weight, and then of course the tumors get big, you give them the GLP-1 agonist, they lose weight, then tumors go back, and they don't grow so fast anymore. You take the GLP agonist away, the mice start gaining weight again, but the tumors still don't grow, right? Or there's at least some element of protection there. So it's going to be interesting to sort of look at that weight loss and weight gain and how that affects these different populations.
So a couple of different questions over here. One is the initial finding that you have with the anti-PD-1 working more effectively, if you will, in the high-fat setting. Have you tried to test that idea in the TCR null mice?
In the TCR null mice?
Effectively, you would be able to...
If we deplete T cells, so we did some T cell depletion studies, and we still had the same effects. Right, actually, the myeloid cells on their own can have some anti-tumor effects. And what we saw is this is not T cell dependent in terms of the myeloid. Most of what we did there was try to see is are T cells important for the induction of the PD-1 of the macrophages, and that's definitely not the case. In fact, if anything, it might be the opposite. But we don't think this is T cell dependent. We think this is just the myeloid. We think they're both inducing PD-1 and using the PD-1 independently. When you block PD-1, of course, you're blocking both T cells and the myeloid cells. So, and the T cells are the main killers, so they're still the effectors. But we think the macrophage population, we think, is an important antigen-presenting population there to help stimulate the T cells in the first place. So they kind of have to go in concert.
Yeah, I think the only reason I bring this up is because when we originally did these under conventionally fed mice conditions, the TCR deficient mice virtually had no anti-PD-1 responses over there. So you would imagine now if you are putting them in a high-fat situation where the macrophages start to drive a response, that would be a great control to tie in response under one setting but not in the other.
We did actually see a little bit of an effect in the high-fat diet when we did our T-cell depletion, but in those experiments, I don't really want to interpret them too strongly because we deplete T cells, it's not like 100%, right? So there might be a myeloid aspect to it, maybe there's a residual T cell component.
Second very quick follow-up is we are seeing, at least in some settings, where we are working on a transcription factor that regulates lipid sensing. I honestly do not know this myself, but looks like fatty acids are a critical precursor for leukotriene production, which could be impacting chemo attraction, immune infiltration.
Sure.
So any comments in that context in your model?
No, I don't have really much to add. I mean, it's a good thought. We don't know what the macrophages, we know that they increase lipid uptake when we have a PD-1 signal. They seem to have a fitness advantage in the presence of hyperlipidemia. Metabolically, what they're doing with it, I have no idea. They could be, maybe that's allowing them to metabolize the lipids, or maybe they're transitioning or converting it to something else. Yeah, I don't know. You'd have to do some interesting tracing to track that down, but it's a good suggestion. Yeah, Gordon. I do not. The question, I'm not sure everybody heard. The question is, how does the PD-1 signal in the macrophages? And it's interesting to think about it because in the T cell case, we think about it as tuning down a signal like the TCR, co-stimulatory signal. And the myeloid cells, we don't know what it's tuning down, or if it's doing something altogether different, right? I would think if it's, functionally it seems to be very parallel, so we think it must be tuning something down, but I'm not sure what that signal would be. And I'm very interested in learning a little bit more about that, but I don't know.
Yes, you also need a kinase to phosphorylate PD-1 for activity.
Yeah.
Is it known what in macrophages would,
No.
what signal would give you that kinase activation
No.
to phosphorylate?
No, that's not known. Again, it kind of comes down to the fact that there's, I mean, you're, of course, world expert in this area. Most of the PD-1 signals that people studying T cells, right? Again, it's known for almost 10 years now that macrophages will express this, but there hasn't been a lot of work done on it. So I don't know the answer to that question. I'm not, and maybe somebody else does, but I'm not aware.
Hey, this is a follow-on to Lisa's question. I'm wondering, have you looked at the effect of cancer cachexia on macrophage function, PD-1? Because obviously low-fat diet is great, but then my patients in the clinic who are losing tons of weight,
Yeah.
I worry that they have some immune dysfunction.
Yeah, this is, again, this is very basic modeling work. The real-life cachexia sort of situations, we haven't not looked at that. I'm not exactly sure, so I don't really, it's a very interesting question to think about. So I think I'm going to have to call the time. I'm happy to talk and answer more questions later on, but I'm getting some signals. So anyway, thank you.